Hi, so we're going to get right into data science today, and my focus is on bioinformatics. And we're going to start with just assigning a quick variable, and what we're going to do here is the purpose of data science and in this case bioinformatics is when you have lots and lots of data. You have thousands or hundreds or millions of numbers of, in this case, letters, A, C, G, and D, or maybe a U or something like that, and or proteins, and you're trying to look for patterns. You're trying to look for maybe a certain sequence in pages and pages of data. And using Python is an excellent way to do that. R is another programming language you can look into as well. So we're going to start with something very simple here. And we're not going to get very deep beneath the surface here. We're just going to be I'm just going to show you a few functions just to get started. So first off, let's say we have some DNA base pairs. I'm going to just make something up here. There we go. So, and if I want to, so just like before, we saw that's a string. And then we can, if we want to print the DNA, I'm going to go ahead and run that. Now, we will be getting into finding the length of DNA, or the length of whatever it is. You can see printed down here, and I'll show you the length in a moment, and then I'm going to show you how to slice, how to find certain segments within that sequence, and then we're going to find out whether something is in a sequence, with the location of that, and then I'll show you how to open and close files. I'm going to focus on text, but you can do it with FASTA files as well. So let's say, so this is, of course, this is too much for me to count. And so I am going to, there's a nice little function called length. This is just my variable here. And it's the length is len and then DNA. It's that simple. And I'm going to go ahead and then print length. That's just a variable that I chose. And we're going to find out what the length of this long sequence is. And it is 39 characters long. I wouldn't have known. I can't count that high. So now let's say we want to break that up. So if there's a certain location in that sequence that you want to break up, we can do that this way. So we're going to do DNA, and I'm going to call this DNA slice, and then DNA, okay, we'll do 10. And what this does, this starts in the 10 from the beginning, 10 spaces in, and then it just prints everything after 10. And so if I do print, you'll see what happens. DNA slice. And there we go. So now you can see we have an incomplete sequence here, but we have basically the second half of that. Or I should say second, last three quarters. Now, I'm going to go ahead and do DNA slice. I'll put, I don't know, first. Can't call it the same thing. So actually, I could call it the same thing. It would just reassign the variable. So we'll do DNA and then colon, we'll just say 15, just to 15, just to make it different. And if I run that, you'll see that it's going to take everything in the beginning up to 15 spaces, up to 15 characters. And, oops, I'm sorry. Didn't do that. Print DNA slice, and there we go. That was just the first 15. Counting from 0, by the way. So the first position is 0, not to be confused with our first position is 1. Now, if we go to, so let's, oh, and then this is another one too. So if we could actually print the entire thing. So you could just do like print DNA and then colon like that. And what that will do is that will, I mean, you don't have to assign a variable every time. I was just doing it out of habit. And if you're writing more code, you always want to use variables. It's going to be easier. And then you can do like print DNA. Again, I can even slice even these variables because they become their own strings. But I can do like DNA, let's say 5 through 25, okay? and then I'm going to get just that section out of the middle. You can also do spacing too, and where you pick out one out of every 5. I won't get into that in this lesson. I want to keep this short. I like to keep these around 10 minutes. So now let's say you want to know if something is in the DNA sequence. So I'm going to look in this DNA sequence up here. Let's say, okay, TAG, okay, TAGT. I'm going to copy that, and let's say we want to know if it's in there. So it is kind of what you think. If TAGT in DNA, remember the if statements from before, then I'm just going to say print, and I'll say, yes, it is in there, baby. And now let's go ahead and run that, and it is in there. It's in there because we know it's in there. I already checked. Now, that doesn't tell you where it is, though. It just simply tells you that it exists. So if you want to find out where those base pairs are, you can do, there is a great little function called find. So I'm going to assign another variable. I'm going to say find DNA. And then you go DNA, that's the file, or that's the string. So DNA dot find, and then let's look up, what is that again? I call it TAGT, okay? So find okay, TH, and then we can do print. We'll say OTAGT. Oh, those windows are always annoying. Oops, is in space, and then I can do comma. Like I think we've worked on this before, and then find DNA. And so that is going to give me the exact location of that, of where it starts. So right there, it's in space number 14. That's where it starts, is in space number 14. All right, so that shows you where you can actually find it. So if sequence, I should also say, and find function. Okay. Oh, oops, I'm sorry, that's right here. So this is the find function. So let's go ahead and move this right there. All right, now is where it really matters, because when you're dealing with bioinformatics, and I'm sure that astronomers are dealing with the same thing, same issue, environmental scientists are doing the same thing, where they have millions and millions of bits of data, and they're not going to sit there and read through everything. So here, one thing that's very common is text files. Text files are, they're small, and they are, when they're big, they're small, and so I am going to show you how to access a text file. So I'm just going to call this my file. Again, I'm using variables when I don't really have to all the time. Actually, here I think you have to. And so we're going to call it DNA text, okay? Nice and simple. And then comma W. That means what I'm going to do is I'm going to write a new file. I'm allowing my software to create the file and write it. So this file does not yet exist, is DNA.txt. And then I'm going to call it my file. And then that would be, my file name, DNA.txt. Then my file dot write. And then, okay, now we're going to say something. I'm going to do, again, make it more base pairs, and then maybe some other text. Okay, and so that is going to go right, I'm creating a file, and that's going to go right in there, and then into that file. And now I'll go ahead and write this. Now you will not see this file. I'm not going to open it for the sake of time right now, but that is, did I run that? I think I did. Yeah, and so what it did is my computer just created one file, and it will overwrite this file every single time with this stuff in it. So now let's say we have to read from that file, because typically you're going to be using a file with a sequence. It could be very, very long sequence, pages and pages and pages and hundreds of pages, and you want to read from that. So how do you do that? I just made a very short sequence. Of course, that's probably like, I don't know, 50 characters or so. But the way you do that is you're going to go My File, just like before, Open, and this is a little different when you're going to read. So DNA dot text, and sorry about those pop-up windows. I'll figure out how to get rid of those later. And then R. So now that allows us to read from the file. And then we're going to do my, let's see here. Okay, this is line. So this is how to read an actual line from the file. So line, and I'll say my file dot read line. That's going to just simply read the first line of the file, and then print line. And I think I've got this all correct here. Let's see if it works. Moment of truth. And there it is. There is our, so what happened is we opened up a file, and then we put stuff in that file, and then we read it. Now, you might be just accessing a file and just reading it. And if you just want to read it, you do this down here. If you already have that file and you just want to read what's in it. And then again, just like what I showed you, you can take a lot of these tools and work them into the opening and closing text files or FASTA files, and that's it. Thank you for joining me.